Okay, so we're recording, so why don't you take it away, Bob?
Thank you. Thanks for the invitation. As I said, this is really a biological project, and it really goes to something that I found very interesting as a biological problem, which is how does genetic information enable life? And this goes back, I think, to Schrodinger and his 1944 book on what is life. And there were a lot of parts to that, but what I think is this is the first time I think where there's an explicit statement that life is a stable low entropy state, but far from thermodynamic equilibrium. I think it would be fair to say that this is not a state observed anywhere else in nature. Um, the other thing about biology is that it requires information. Um, Life requires heritable information in the genome. Um, and this information is essential for maintaining this living state, this thermodynamic non-equilibrium state. Um, and importantly, the non-equilibrium state allows death. In other words, because it's a non-equilibrium, the living system has to maintain that state using information and acquired energy. Um, so two of the three necessary components of, uh, of of evolution are part of this. One is that the genome permits transgenerational information of information, and the non-equilibrium state permits death, which is necessary for evolutionary selection. But that said, there's really no clear way to link the two. The human genome contains perhaps 30 billion bits of information, but of Shannon information, but that's really just a measure of complexity. Shannon information, of course, does not say anything about meaning, nor does it say anything about mechanism. And I think that the sort of difficulty here is clear in unit matching. For example, the Shannon information is unitless. It's basically bits. It's basically comes out of probability functions, unlike entropy, which is in units of joules per degree Kelvin, and so you can't physically match them. Um, so this was the question: how does genetic information enable the thermodynamic state that's unique to life? And I'm sorry, I have to get to the actual topic. I have to go through all this. Um, the this pro I what I used is the genetic information in enzymes to think about how information works. Um, in in the I'm sorry, reactions in the absence of enzymes or catalysts is governed by the empirically derived Arrhenius equation, which links the reaction rate to the energy of activation and the temperature. Um, Living systems, because they're confined to a very narrow temperature range, cannot use an increased temperature to accelerate the critical reactions, and so they use the enzymes, which is shown here, that essentially capture substrate, and they promote the reaction. They accelerate the reaction up to 15 orders of magnitude. Um, we were all taught in high school that the role of enzymes is to decrease the energy of activation. It doesn't substantially change the thermodynamics of the pre-reaction, post-reaction changes. It simply accelerates them by this peculiar kind of decrease in the activation energy. And in prior studies just pointed out that the value of the genetic information in terms of the thermodynamic value is expressed in this decreased activation energy. That said, although it's captured in the Arrhenius equation as this decreased activation energy, the actual mechanism is at a quantum level. Essentially, the enzymes capture the substrate, they optimize their quantum interactions to increase the probability of a reaction, so that the speed is really, I think, what is the manifestation of this increased probability. So what's been shown is that although basically the structure of the enzyme, which is encoded in molecular at a molecular level, in other words, it's the nucleotide, the triplet nucleotide that specifies the amino acid sequence of the polypeptide. It has to function at its activation site at a quantum level. The Nobel Prize in Chemistry in 2013 was awarded based on those kinds of observations. I think from a just sort of broad point of view, this is a fine graining. In other words, molecularly encoded genetic information has to interact at a finer grain at a quantum level, and so it's kind of the opposite of coarse graining, which is used so commonly in science. And the question is, how does life increase the information content in the genome to do this fine graining? So coarse graining, which is what we think about a lot, involves smoothing of detailed structures and makes this more tractable, but at the cost of losing information and losing entropy. The opposite requires information, and this has to be information gained from the genetic information. So how does that happen? And my proposal is that this occurs during protein folding. Again, is up on the upper left, the sequence of amino acids, which is a linear string of these. It comes from the linearly encoded triplet nucleotide sequence in the gene. But this does not function. It cannot function until the protein folds into a three-dimensional structure. And to me, this is a dimensionality change. So you go from one dimension essentially to three dimensions. The process of folding, if it's controlled simply by the thermodynamics, is based on the interactions of the charges in the amino acids. And it's been shown, this is Leventhal's paradox, that a very large number of potential shapes can emerge from even 100 amino acids. And that is probably the smallest size of an enzyme. Many enzymes have 2,000 amino acids. What Leventhal calculated, and I still find this hard to believe, is that this huge number of potential shapes are available. And so, again, just looking at it from a thermodynamic point of view, you could say that there are a large number of potential three-dimensional structures, each of which would have a certain probability of occurring, given just purely thermodynamic control. But what's interesting is that cells actually use an extensive molecular machinery, and I think that's here, to control the kinetics of protein folding. And so rather than this purely thermodynamic state, which is shown here, that the ribosomes and the chaperone proteins, so the human genome has 80 different ribosome proteins, and probably a larger number of chaperone proteins. And this kinetic control then makes with very high probability that the end result of the folding process will be the functional three-dimensional structure that's needed for the enzyme to act. This is a highly improbable outcome. And so I think that if you just look at this, I'm sorry, as a Shannon information process, where now you sort of say, what are the possible outcomes in terms of three-dimensional structures? What are those probabilities? And because the cell controls the kinetics of information with a very high probability, for perhaps 90% of the time, and maybe more, this one particular three-dimensional structure emerges. This is the one that's functional. And so in doing that, I would propose, and this is honestly a new concept, is that the genetic information is actually expanded through this folding process. And so the thought here is that the dimensionality change, going from one dimension to three dimension, and having that as a controlled process represents a gain of information. Now, how does that actually work in terms of the thermodynamics of the cell? And here I go back to Maxwell's demon Gedanken, and I think probably everyone is familiar with this, but just to clarify, Maxwell had a thought experiment. He called it a finite being, but ever since then it's been known as Maxwell's demon. But this finite being, and you start with a two connected gas collections. The demon can watch the gas molecules as they approach a frictionless door. The demon opens the door if it's a fast-moving molecule, closes it if it's slow-moving. So essentially collecting the fast-moving molecules on one side, the slow-moving on the other. And in Maxwell's thought experiment, this is a violation of second law of thermodynamics. And that has been, I think, how it's been looked at for the past 150 years, based on the literature I can read at least. But to me, I think that the lesson here is one of fine graining and coarse graining. We typically view the Boltzmann distribution as linked to the temperature, and in a way that we say if we increase the temperature, the Boltzmann distribution changes. What the demon does, I think, is change the Boltzmann distribution, resulting in a visible change in the coarse grain appearance of that, which is the temperature. This is not a violation of the second law of thermodynamics, but it is an interesting way to interact at the interface between the macroscopic and microscopic system description. And so if we think about how the enzyme acts, I think that also by acting at that same kind of interface, in this case, the collective function of the enzymes is to create a substrate and product concentration that's identical to what would be seen in a higher temperature state, but in the absence of a heat influx. And so I think, like the demon, the actions of enzymes produce a non-equilibrium thermodynamic state. In other words, the concentrations of substrate and products has created is one that would have been there if the temperature was very high. But in the absence of that, that will continuously deteriorate without a continuous upgrade, a continuous action of the information and the necessary energy to generate these reactions. Again, not a violation at all of second law of thermodynamics, but an interesting interaction at the interface of the molecular of the statistical physics and the thermodynamics. And notice here now, we have the statistically encoded Shannon information acting not on the thermodynamic states, but on the statistically based microscopic states. The statistical probability of a reaction occurring at a quantum level, and the statistical physics of the Boltzmann distribution. So it's that interface, I think, that's important. So anyway, that's I hope that's at least reasonably clear. But that's how I think genetic information allows life to occur. And this is just an example of this. Again, if we go from substrate to products, and you get a change, you can either increase the temperature or you can add a enzyme, and so you get the equivalent outcome, but the thermodynamic state is different. And because of this non-equilibrium state, failure equals death, and death equals evolutionary selection. And so this is the equivalent. Now, what this model suggests is that evolution is not quite the same selection forces that is typically viewed, in that it's selecting genes. Because what evolution is selecting gene function, the survival is based on gene function. What evolution is selecting is not only the gene, but the molecular machinery that ensures that amino acid sequence from that gene is folded to the three-dimensional structure that's functional. And so, again, this sort of dimensionality change is very critical, and is also under selection by evolution and subject to evolutionary optimization. Okay, so all of that then has led to a hypothesis that the change in dimensionality, this strategy to increase information through dimensionality changes, through controlling dimensionality changes, I think gives a potential theoretical basis for the role of multicellularity. Again, a fertilized egg gives rise to a complex human structure. So in theory, the amount of information in the egg gives rise to the body, but there's a mismatch. I mean, the human genome is very large. It's 30 billion bits, but the human body contains 36 trillion cells, and that requires an enormous expansion of information. And so where does that come from? And so this dimensionality hypothesis suggests that the proliferation of the fertilized egg is a process that is tightly controlled by cell-cell interactions, so that each state there is a highly non-random three-dimensional structure, and that by specifying that configuration out of all other possible configurations of proliferation of the cell, that there's a consistent gain of information, and so that this highly non-random three-dimensional this change from the point source where the fertilized egg is used as a point compared to the three-dimensional structure of a multicellular organism, represents a gain of information that is due to this dimensionality change that is controlled at a local level. And I contrast this to cancers, in which the cancer cells are proliferating, but in the absence of the cell-cell interactions that produce a highly non-probable and complex structure, that cancers produce really structures with low order by comparison, and this represents kind of the dichotomy of these kinds of informational dynamics. So all of which is then a long run for a short slide, and that is I have periodically attended and have tried to consistently view the videos of the lectures that are given to your group on the human consciousness and that sort of thing. Um, what I have thought before this is that you can view information as sort of the critical component of evolution, and that that what's being selected is that there is optimal gathering of information about within the organism about its environment, and that can be both hazards and opportunities, and it doesn't have to be all information, and it's subject to a cost-benefit ratio. But in general, individual organisms, for example, need to constantly search their environment for hazards and for opportunities. Those that do this best and with the highest degree of efficiency win and are being selected by evolution, while those that do not lose out and die off. I would argue that human cultures, at least initially, the focus was on survival, and that, however, subsequently, the information gathering by humans is beyond the evolutionary imperative, meaning that humans use resources to investigate informational topics such as philosophy and mathematics. They look at the universe, they look at quantum scale interactions that have no immediate benefit for survival and proliferation. These are topics that go beyond the evolutionary imperative, and to me is what constitutes consciousness. And so within the hypothesis of information expansion as the result of dimensionality changes, it occurred to me that this could represent a similar process here now, because humans uniquely in the animal kingdom can access and use time. So there's an additional dimensionality that's been gained over time, and that because humans recognize time as a sort of non-spatial dimension and can, for example, measure time and write equations that successfully predict time-dependent dynamics within the spatial dimensions, that this expansion of dimensions is something by which evolution allowed human consciousness to emerge, and that its location is in some way based in the dimension of time. So I passed this on to Ken as just for his thoughts, and he thought it was interesting enough to present this. I will say that this, although the article that I've prepared for this, the question of how does genetic information enable life, I have used the three-dimensional gain as part of an expression of multicellularity. I have not added anything about human consciousness, because I thought that the probability of the paper getting accepted alone is pretty low, and if I had human consciousness, it would basically end all probabilities. But I hope we're among friends, I thought it would be reasonable to at least try to kind of summarize my thinking about this and how this emerged from a purely biological analysis. So with that, I'll shut up.
Oh, thank you very much. We'll take questions and comments now. If you have one, make sure you unmute